97000 to this is 1 Now, and we all know this paper is for 1 hour 15 minutes and this is for 60 marks. Please remember that the time is very little and you need to really concentrate on the questions and understand the questions and then start answering them. Question number 1, figure 1.1 is a diagram showing part of a cell membrane of an animal cell. And you can see state the approximate thickness of the membrane, anything between 5 to 10 nanometers, but we write the units when nanometer. You can't say micrometer. So you can also say a suitable range within the range of 5 to 10 nanometer. Unit must be shown. You must show the unit. It is very important. So 7 nanometers. Complete table 1.1 to show the names and functions of the components of the cell surface membrane. The letters of the labels in figure 1.1 that identify each component. So channel protein, what is the function of it? Facilitated diffusion. So facilitated diffusion, and the letter on figure 1.1 was A. You can see here A is the one which will allow facilitated diffusion from say 10 to 5. If something is 10 here, 10 potassium ions or sodium ions, and 5 here, so it will be facilitated. Right? What is F? Cholesterol. B is a glycoprotein. C is a channel carrier protein. D is a glycolipid, and E is a phospholipid layer. GH is the entire uh, phospholipid bilayer. Then which says phospholipid, what would be the phospholipid? Would be E. Then what is the function of the phospholipid? It will be forms a bilayer, you could have said, or you could have said it's a barrier to water soluble and polar substances, because this hydrophobic part is a barrier to polar molecule. This whole thing is a barrier to polar substances. It will allow non-polar substances to pass, allows fluidity, provides stability, and forms a hydrophobic core. These are all the words that you could have used for this. So phospholipid, then receptor for cell signaling. You would say A. Sorry, no, you won't say A. You would say glycolipid or glycoprotein, because here you have to say the component. So you could have said glycolipid or glycoprotein. And here, receptor for cell signaling. Let's look at that. It could be B or D. It could be B or D. So B is this, and D is this. Receptor for cell signaling. So you could have said glycolipid or glycoprotein. Then the last one is, letter on the figure is F. What is it? This is cholesterol. And what is gives stability, regulates fluidity. These are the words that we use. So once again, now that I've written all of these, so channel protein facilitated diffusion A, then phospholipid is a barrier to ions, or you can say it's a barrier to water soluble substances. You can say it's allows diffusion of non-polar substances, allows fluidity, provides stability, forms a hydrophobic core. All those things you could have said. Then glycoprotein is a receptor for cell signaling B or D. You have to write, in fact, both of them, B and D, to get the mark for that. Cholesterol gives stability, and that was F. So 4 marks for these 4 lines. Now, coming to part number B, figure 1.2 is a drawing of a transmission electron micrograph TEM of a cell from the palisade mesophyll of a leaf. The drawing does not show all of the organelles visible in a transmission electron micrograph. Complete figure 1.2 drawing and labeling a mitochondrion, rough endoplasmic reticulum, smooth endoplasmic reticulum. Your drawing should show the detail that can be seen in a transmission electron micrograph. Now we need to do the drawing. This is for 3 marks: a mitochondria, rough endoplasmic reticulum, and smooth endoplasmic reticulum. Now, how would you get your 3 marks? Mitochondria with 2 membranes, at least 1 cristae. Rough endoplasmic with at least 1 cisternate, 2 lines close together and ribosomes attached on the outside. Smooth endoplasmic reticulum with at least 1 tube with no ribosomes. So if you labeled it RER, SER, that was acceptable as a label. RER should have been attached to the nuclear membrane, and SER can be anywhere. It's not necessarily attached to the nuclear membrane. Identify the organelle labeled X, Golgi body, but what is it called in a plant cell? Golgi body is called, yes, fine, but there's another name for it which I always tell you because it has come in one of the papers as well as Golgi body, and the other name for is, you could have said Golgi apparatus as well, but the other name which you must know is a dictyosome. Both were allowed. I'm not saying that you have to write dictyosome, but if then dictyosome comes in an MCQ and you don't know that, that's going to be sad. So modifies proteins. Function is modifies. Please remember to learn the correct English. Modifies proteins. You could have said modifies proteins. There's only 2 marks for that. So modifies proteins, you could have said that. You could have said packaging of proteins. You could have said forming lysosomes. So 3 possible options. You could have said modifies proteins. You could have said packaging of proteins into Golgi vesicles. And you could have said forming lysosomes. So 3 possible answers with only 2 marks. 1 mark for this and 1 mark for this. Question number 2. Water is the main component of blood. Explain. So if it's an explain, it's a lot of bio. Explain how the properties of water make it suitable as the main component of blood. So water is a good solvent, 1 mark. Transport of urea for excretion, or you can say transport of glucose. So 1 named substance and a correct reason. So transports urea for excretion, you could have said that. Then it has high specific heat capacity, and the amount of heat that must be added to 1 unit of mass of the substance to increase the temperature by 1 degree Celsius. Now, high specific heat capacity, of course, you've given the definition. That gets you a mark, but then if you tell me, so temperature of the blood remains fairly constant and heat is dispersed throughout the body. That's a lot of English. I must tell you, your English must be very good. Or you could have also said water is a dipolar molecule. Dipole or dipolar. So there were 4 5-mark scheme points, and any 3 of these, and you would have got your 3 out of 3, which teaches you to write more so that you get your 3 marks. So water is a good solvent, transports urea for excretion, high specific heat capacity, or if you gave me a description of that, the amount of heat must be added to 1 unit of the mass or 1 gram of water to increase the temperature by 1 degree Celsius. So that is important. So the temperature of the blood remains constant. Temperature of the blood remains constant, not body. Or you can say heat is dispersed throughout the body. And water is dipolar. Then it says figure 2.1 is the diagram of the circulation in a mammal. Complete by naming P and Q. What is P? P is the aorta. Why? Because the aorta comes out of the, you can see this vessel is coming out here of the left ventricle. So coming out of the left ventricle, what is Q? Q is the vena cava. We are not interested in you telling us superior, inferior, because this is a diagram. It's an artist's art diagram as a depiction of the 2 pulmonary and the systemic circulation. So inferior, superior didn't matter. You just had to say vena cava. So this was, as you can see, this is only for 1 mark. This was a very easy basic. This is for O levels, in fact. So it's only 1 mark. The mark tells you there wasn't a very A-level skill, it was an O-level skill. So they're only giving you, if you, either you get 1 mark or you don't. There's no 0.5 in bio, please remember that. Then the part 2 of the question, describe the functions of P and Q. So what are the functions of P and Q? What is the function of the aorta and the vena cava? So you could have said aorta transports oxygenated blood to the body, got a mark for that, or you could have said aorta transports blood at high pressure to all the organs of the body. That's the same mark scheme point. The second mark scheme point would have been vena cava transports the deoxygenated blood from the entire body to the right atrium. Or you can say vena cava transports blood at low pressure to the right atrium. So any of those points, as long as it sort of fitted into the functions of P and Q. You could have said aorta transports oxygenated blood and vena cava transports deoxygenated blood if you did a comparison. And you could have said another comparison, blood in the aorta is to the body or the respiring tissues, and blood in the vena cava is returning back to the right atrium. So a lot of possible answers, but easy to get these 2 marks, even if you wrote a lot. But if you got the message across to the examiner, that was good enough. So aorta transports oxygenated blood to the body, vena cava transports deoxygenated blood to the right atrium. And I've given you the other alternatives as well. Aorta transport oxygenated, vena transports deoxygenated, blood in the aorta to the body, blood in the vena cava to the right atrium. Part 3 says explain why the mammalian circulation is described as a closed double circulation. Now that's come in millions of other papers as well. Closed means it is blood is contained within vessels. Double means blood flows twice through the heart in one circulation. Or you could have said there's a pulmonary and a systemic circulation. So simple English, simple basic way how you say it. Closed blood is contained with vessels. There are some animals which have an open circulation. Then you should be figuring that out. Maybe you should Google that. Which animals have an open circulation? And which animals have a single circulation? They don't have a double circulation. Fish has a single circulation. So then look at the opposite of it, and then maybe you can learn a little more of biology and be a little more interested. Gen Z needs to be a little more interested in biology. I like the word Gen Z. So the headings you need not have written. I've given the headings. Closed means blood is contained within blood vessels. Double means blood flows twice through the heart in one circuit, or you could have said one circulation. You could have named it also pulmonary and systemic circulation. Then we come to the next question, part C. Figure 2.102 is a transmission electron micrograph of a cross section of an arteriole. Blood flows from muscular arteries through arterioles into the capillary network. The lining of the arteriole is folded because the arteriole has constricted. This constriction causes the blood pressure to decrease from 12.7 kilopascals in the muscular artery to 2.7 kilopascals at the end of the arteriole. Now you can see the electron micrograph, and it's telling you how the muscular artery first, of course, will enter, and then it's going to divide into arterioles, and then capillaries, and then venule, and then vein. And also look at the magnification, it's 2000 times. Explain why it is important that the pressure of blood decreases as it passes through the arterioles. Now, you've got to understand why would it decrease. Blood at high pressure will burst or damage the capillaries. Capillaries have thin walls. They're only one cell thick. And low pressure blood flows slowly to allow exchange or the diffusion of substances. And pressure will be low at the venous end of the capillary, so the tissue fluid is reabsorbed. So all those points you have to give me for why it is important for the pressure of the blood to decrease as it passes through the arteriole. So you've got to understand, as I explained here, this would be the artery, and then this would be the arteriole, and then it's the capillaries. Capillaries or walls are one cell thick, or you can say they have thin walls. So blood at high pressure will burst or damage the capillaries. So capillaries have thin walls, or capillaries have one cell thick wall, and they're made up of one layer of endothelial cells. Please remember the word endothelial. At low pressure, blood will flow slowly so that exchange or diffusion of substance can happen in the capillaries, where is the only place where exchange of substance can happen is in the capillaries. Pressure will be low at the venous end of the capillary so that tissue fluid is reabsorbed. So high pressure will burst the capillaries. Capillaries have thin walls. Low pressure blood flows slowly to allow exchange of substances. Pressure low at the venous end of the capillary so tissue fluid is reabsorbed. Then it says compare the structure of muscular artery with the structure of the arteriole shown in figure 2.2. Now, when the comparison of the muscular artery to the structure of the arteriole, the basic similarities were both have endothelium. Because it says compare. Compare means similarities and differences. So both have endothelium, both have tunica media or smooth muscle in the wall of them. This wall of these blood in the wall of these blood vessels. So arteriole, muscular artery, both have endothelium, both have tunica media or smooth muscle. Difference is muscular artery. So let's talk about the muscular artery, and we give the heading muscular artery. Muscular artery has a wider lumen, has more smooth muscles, while thick layer versus the thin layer of smooth muscles. Or you could have said a thicker tunica media. Then you said more elastic fibers. Then you could have said more collagen fibers. So these are the things that you could have mentioned in this question, which is of course a 3-mark question. So these are all the points that you could have given me, which I have. Of course, you can pause the video here and have a look at what I've worded it. So the muscular artery and the arterioles, either comparing the arteriole with the muscular artery. So of course, you've got to understand muscular artery is the one which is entering the organ, and that's going to divide into arterioles. So things are going to diminish, smooth muscles and all. There'll be more smooth muscles in the muscular artery. There'll be more elastic fiber in the muscular artery. All these differences I'm talking about is the muscular artery. So you must give a heading. Whenever you do a comparison, remember to say, you can't say it has. You're talking about the muscular artery because it's mentioned first. The muscular artery is being mentioned first. So please, that's why we are allowing you to talk about that first. It's always in the mark scheme if it's in the comes first in the question. Like you say, compare Karachi with Lahore. Then Karachi I've said first, so tell me things about Karachi and then compare it with Lahore. Now, coming to question number 3. A class of students was studying the feature of some human pathogens. One of the students constructed a flow chart to identify 4 different human pathogens. The student used information about the structure and mode of transmission of these pathogens. Figure 3 shows the partially completed flow chart. Pathogen has a cellular structure. No. Pathogen is transported in body fluids, HIV/AIDS. Then pathogen has a cellular structure. Yes. Pathogen is a prokaryote. No. Pathogen is yes. Pathogen is transmitted by fecal oral route. Yes. So what are we going to write here? Vibrio, cholera, and cholera. Because that's what it gives in brackets. Like here. Like here. So Vibrio, cholera, and cholera. That is going to get you 1 mark. Then here pathogen is a prokaryote. No, pathogen is transmitted by a vector. Here you could have also said Anopheles mosquito. And then what is the name of the pathogen is Plasmodium. Plasmodium falciparum. You could have said Plasmodium falciparum ovale or malariae or vivax. So you could have written vivax. I like the word vivax. So you could have said Plasmodium falciparum, you could have said ovale, you could have said malariae, or you could have said vivax. So you got a mark for that. Pathogen is transmitted by fecal oral route. We've done that. No. Pathogen is transmitted by airborne. Or you could have said droplet infection. Or you could have said droplet infection. Fine. You could have said aerosol as well. And here, of course, it would be Mycobacterium tuberculosis. Or you could have said bovis as well. So you could have said bovis as well, or any one of this would have been fine. So this would get you 5 marks. You can see the 5 boxes, 5 marks. Coming to the B part of the question, HIV has a nucleic acid core of RNA. The virus also contains the enzyme reverse transcriptase. After HIV enters T lymphocytes, reverse transcriptase catalyzes the formation of DNA using activated DNA nucleotides with a viral RNA as a template. Some drugs, such as tenofovir, have been developed to inhibit the action of reverse transcriptase. The structure of tenofovir is similar to the structure of deoxyribose adenosine monophosphate, as shown in figure 3.2. After tenofovir is absorbed into cell, it is phosphorylated twice and can be used by reverse transcriptase in the synthesis of DNA. When a tenofovir molecule is added to the DNA strand being synthesized, the process stops. Suggest the mechanism of action of tenofovir to prevent the synthesis of DNA by reverse transcriptase using the information in figure 3.2 to get to your answer. Now, what could have happened? Why is that enzyme being inhibited? So we've got to really figure this out and know our chapter on enzymes. Now, the first and the most obvious thing should have been with you is tenofovir competes with the DATP for the active site. Tenofovir competes with the DATP. This we can shorten it to DATP. But this is deoxyribose adenosine monophosphate. Deoxyribose adenosine triphosphate, because that's been mentioned here that we can talk about that. It contains using activated DNA nucleotides. So it is going to compete with that. Then tenofovir forms a phosphodiester bond to the elongating DNA strand but stops further reactions. It forms a phosphodiester bond to the elongated DNA strand but stops further reactions because it says it's telling you in the question have been developed to inhibit the action of reverse transcriptase. And then they've told you reverse transcriptase is going to make viral RNA, is going to convert the viral RNA into viral DNA. And tenofovir has no 3-OH. There is no 3-OH, so next nucleotide cannot form a phosphodiester bond. Or you can say no deoxyribose, so cannot form the phosphodiester bond. There's no deoxyribose. So no phosphodiester bond can be formed. See here it says after tenofovir is absorbed, instead it is phosphorylated twice, it can be used by reverse transcript. So that is why deoxy monophosphate becomes deoxyribose adenosine triphosphate. It's telling you in the question it can be used by, so it is phosphorylated twice. So from monophosphate it becomes triphosphate. So just read through this and see that you've understood it and you know how to word it. Pause the video here. Part C of the question, pre-exposure prophylaxis, PrEP, is the use of therapeutic drugs to prevent the replication of HIV in the body following infection. The drugs are taken by people who are at risk of becoming infected. Therefore, is one of these therapeutic drugs. In 2016, the United Nations set a global target of 3 million PrEP users by 2020. Table 3.1 shows the number of people across the world who received a therapeutic drug for PrEP each of the years between 2012 and 2019. Calculate the percentage of people who received PrEP in 2019 as a percentage of the target set by the UN in 2016. Give your answer to the nearest whole number. Oh, that was simple because it said 3 million. But in 2019 we only have 605,000. So divided by 3 million, which is into 100, and you get 20%. PrEP does not prevent transmission of HIV. State and explain how health authorities can reduce the transmission of HIV. Whenever it's a state and explain, so state means you just state, explain means you give a biological explanation to it. So there must be at least one statement or at least one explanation to get the maximum 4 marks. If you just state things, then it's not going to get you your 4 marks. There are 4 marks for this. Everybody must get their 4 marks. Very simple question. Supply condoms, 1 mark for protection during sex, barrier to transmission during sexual intercourse. That's an explanation for that. Needle exchange schemes, decrease risk of sharing contaminated equipment, use new needles or sterilize equipment for medical procedures, decrease risk of transmission for contaminated blood, provide testing for HIV in high risk groups so that early diagnosis or newly infected people can start drug treatment immediately, test pregnant women for HIV to prevent women who are HIV positive passing the HIV in breast milk, prevent people who are HIV positive being blood donors, prevent people receiving blood infected with HIV during blood transfusion, carry out contact tracing, locate people who are undiagnosed, maybe HIV positive, should be offered the test, supply drugs to people living with HIV, prevents HIV spreading throughout the body, then provide education about HIV treatments, raise awareness of ways to reduce infection, and uh, and make it a law to make it illegal to knowingly transmit the virus. So lots of points, and any four of those were fine, and you would have got your 4 out of 4. Uh, there's a whole uh, there's a whole about 18 mark scheme points. Please look at the mark scheme for this. I'll put it at the end of this video, and you can have a look at it. So supply condoms, barrier to transmission, needle exchange, decrease risk of sharing contaminated equipment, use new needles, decrease risk of transmission from contaminated blood. So you had to get your 4 marks. So that was an easy out of about, I think, 18 mark scheme points. These are all the mark scheme points which are available to you. Please go through all of them and just read through them and remember them if a question comes on this sort of a question comes again in the exam.